Professor Ron, can we talk about someone approaching a crime scene and collecting fingerprint evidence? Who would that person be and how would they approach this evidence?
The person that it would be is what we term a criminalist. They are the ones who they study criminalistics. They are the evidence collection team. Everything that is collected, not just fingerprints, but swabbing for blood, semen, saliva, perfumes, we can do scents too. These are done by criminalists. Let me explain it quickly, if I may. There's three types of forensic scientists, if you will. There is the front line, which are the criminalists. They go out and collect and preserve everything. Then there's the laboratory criminalist that runs the assays and runs everything through the equipment, through the gas spectrometer and through microscopes, all that stuff. That's the lab technician. The third level is the scholar practitioner that writes the algorithms, if you will, for that the laboratory people would use to say Armstrong 2013 states that this person is 5'6" to 5'9" because ... I myself am a forensic practitioner. I do research and I teach, and I hope to create out of my research an algorithm one day. But to answer your question specifically, it is a criminalist that does all of the work, and they are certified. It is not an easy topic to do, to roll prints, because there's rolling live prints, rolling prints off of a decedent, rolling prints off of a decedent that's been in water. There are all sorts of techniques and ways of rolling a print, and that's just collecting prints from a hand. Then there's also finding a print, which we call a latent print. Latent means hidden, basically. So there's finding the prints and developing them, brushing things with the powder and the brush and all that stuff. There's so much to just a fingerprint or a thumbprint or an ear print or a footprint. We have ridges and valleys all over the place, and we have oil all over the place and proteins, and you can get things off of this. And because this is for writers, I'll give you a trick. A bloody fingerprint. Do you do the blood, or do you develop the print? Blood gives you DNA and other, do they have STDs and stuff like that? And now with a human genome mapping, God knows what else they can find out from it, depending on how big the sample is. Whereas working with a fingerprint gets you into APHIS, and the fingerprint database is getting bigger and bigger and bigger and bigger. But so is the DNA database. What do you do? I'll let you decide.